Hello, everyone. I am Ayush Raman, and the title of my talk is Decoding Biases, Hypotheses, and Regulation in Big Genomic Datasets. Here is my introduction. I'm a computational biologist at Broad Institute, and I'm interested in understanding the dynamics of gene regulation using data integration. I develop tools and approaches for data analyses, such as distinguishing biological signals from latent variables or noise, such as batch effects, and apply machine learning algorithms to understand biology, such as elucidating intratumor variability or clonal variations in iPSC cells using single cell genomics. My background is as follows. I did my engineering in bioinformatics from Vellore Institute of Technology, Vellore, Tamil Nadu. Master's in computational biology from Carnegie Mellon University, Pittsburgh, Pennsylvania, and my PhD in quantitative and computational biosciences from Baylor College of Medicine, Houston. This is the summary of my background work. And as we all know, there has been a massive influx of big data in the field of genomics, and hence to gain knowledge or biological insights, one needs to first detect the biases or the batch effects within the data set, which can creep up due to the inherent technology or basically generating the data across different batches from different days or using different personnel. So to this end, my first work was developing a universal batch detection algorithm to detect noise or batch effects, which can arise due to sequencing of samples in batches, and statistical approach to detect errors due to PCR amplification. Now, now the second work of looking into the biases of due to PCR amplification, there was a paper that was published in Nature 2015 that shows the pathogenesis in Rett syndrome, and we showed that the mechanism what the authors showed can be an artifact of PCR amplification. This paper was published in Nature Communication. Now, these were two cases of intersample variation. Now, to next understand the biological variation, I was interested in looking into the intertumor heterogeneity. We showed that STK27AC, which can be an inactive enhancer mark, can distinguish between the stages of progression and can also define epigenetic subtypes that were correlated with transcriptomic subtypes. Next, during the course of my research work, I've published about eight first and co-first author papers across high impact journals, and have been fortunate to be awarded for my research as Junior Research Parasite Award for rigorous secondary data analysis and MC Bio's Young Scientist Award. I believe in collaboration and have worked in big data consortiums such as TCGA, and have a background in teaching and mentoring students. I have also reviewed manuscript in major journals such as Computational and Structural Biology, Stem Cell Therapy and Research, and IUBMB Life. Beyond research, I'm interested in teaching. I've taught and graded and prepared assignments for courses in computational biology, applied machine learning. I've taught a course on ChIP-seq data analysis at Institute for Systems Biology for their nucleic acid course. And I've formed a next-gen sequencing club at ISB, where we discuss molecular biology techniques and papers and computational methods for data analysis. I've taught I've taught coding and scripting to my wet lab colleagues, both men and women, and believe in equality and STEM education among young girls and women. I'm highly interested in returning to India for an independent research career after spending 15 years in US. And the insights that I want to gain from this meeting is to know about the paths and prospective in institutes where I can be a good fit, paths in terms of scholarships and fellowships. And if anybody is interested in collaboration, or basically if their research interest aligns with mine and my skill sets can be complementary to this, please contact me via mail, or you can DM me on Twitter or through LinkedIn. Thanks a lot. You have a nice day. Bye.